Well, we are officially back, and we're getting ready to really encroach into this area of understanding how some of the forensic and personal genomics is actually done. In other words, we now know exactly what is measured, but how do we apply them to populations? And we really find ourselves in a position where we have to go back for a moment and review a little bit about how geneticists deal with populations. And you can kind of see here and guess. It's rather timely, as one of our last exercises in lab is usually dealing with Hardy-Weinberg equilibrium. So I get a rare opportunity here to talk a little bit about a slightly different perspective than you may have heard before about Hardy-Weinberg equilibrium, and also to give you a couple examples and a little more detail into the background of it. Of course, the traditional story, if you will, is that in 1908, Weinberg intuitively hypothesized that there's a relationship between alleles or genes and genotype. Remember 1908? We're in a position where Mendel's ideas have been accepted in the scientific community. So we know that certainly among eukaryotic organisms, we know we have diploid organisms. So we know that there are two alleles at every locus. Sometimes you have to be a little bit careful of the terminology. Remember, when we talk about genes and alleles, they are the same thing. Genotypes are the combinations of the two alleles or genes in diploid organisms. So very often you'll see it described as the relationship between genic and genotypic structure, or allelic and genotypic structure. So genic and allelic, again, synonyms of one another. The ideas that were proposed by Weinberg were really formalized by Hardy, who was a mathematician, and eventually we became known as the Hardy-Weinberg equilibrium. Of course, the story itself and the history background is really much more complicated. Mendel's work, as you know, took between 40 and 50 years to come to fruition, in the sense that the scientific community adopted the idea and realized that Mendel was correct. However, there were a lot of people that were still arguing in the 1900s that environment was much more important than particulate inheritance. And one of the real champions of anti-Mendelism, if you will, was Udny. Love his name. I hope I'm pronouncing it correct, but Udny Yule. And he said Mendel could not be correct because he thought that over time, any alleles that showed dominance in a Mendelian concept would eventually overwhelm any of the recessive alleles and always continue to increase in frequency among populations. In a bit of time, one of the great champions of Mendelism, William Castle, suggested that in the absence of selection for one or the other of the alleles, genotypic frequencies would always remain constant. And one of our other buddies, Carl Pearson, remember chi-square analysis, Pearson correlation coefficients, all these individuals were really involved in the early 1900s to try to define genetics as we know it today. And Carl Pearson was more of a mathematician, obviously, being R.A. Fisher's student, and he found that there was one position of equilibrium, and that's where P, or the genic or allelic frequency of one allele, was equal to Q, or the genotypic or allelic frequency of the other or alternative allele, when they were both equal to 0.5 or 50%, okay, then the genotypes would remain constant. And of course, this is where we find, the entire idea, if we were to take a peek and to say, oops, my pen's not working. Here we see the classic P plus Q equals 1. And of course, if we expand this to include the matrix of genotypes, where we see, and you all remember this, I'm sure, P squared plus 2PQ plus Q squared equals also 1. We see that the P squared, which is equivalent to kappa genotype, PQ is equivalent to the kappa small a genotype, and Q squared is equivalent to the small a homozygous genotype. So the ratios are 1 to 2 to 1. So this was really Carl Pearson and William Castle contributing this idea that this would be proof that, of course, the Hardy's idea and Weinberg's idea was substantially correct. However, Ewell changed his argument a little bit and said, well, okay, if P equals Q equals 0.5, perhaps that would be the only condition in which the genotypic ratios would remain stable. He said, well, it would not occur in any other condition, because under any other frequency of alleles, dominance would overtake the recessive allele. Punnett had no way to argue, and in fact, Punnett played on a cricket team with guess who? Hardy, who then walked in and wrote a single-page letter to Science magazine. The text is right here for you. I copied it out of his letter. Go ahead and please pause the video at this point and take a peek. Read the letter, because it's really quite an interesting view into how people wrote and how people thought back in the 1900s. And his question really arises down here in the final. He says the question is, in what circumstances will this distribution be the same as it was in the generation before? And again, he says, well, it's really easy to see under standard conditions where P equals Q equals 0.5. However, he furthers the postulate by saying that no matter what the frequencies are, the distribution should be unchanged after the second generation. So really, this was Hardy's response to Ewell, and Ewell basically conceded at that point. So Weinberg was known as Hardy's law at the time. Kurt Stern, a geneticist from the 20th century, a little bit later on in the 1900s, really was the one who said we really should add Weinberg's name, and in fact even suggested it should be Hardy-Weinberg-Castle equilibrium, because Castle worked out the ratios for the simplest of cases, as I mentioned before. Ultimately, the genetic community really accepted the idea. Hardy-Weinberg-Castle became just too big, and there is some historical notes that maybe Stern wanted his name kind of attached to the idea of Hardy-Weinberg equilibrium, but certainly everybody came up suggested, well, okay, Hardy-Weinberg is really the best name for the mathematical principle. And really simply stated, it's just that genetic variation in a population will remain constant from one generation to the next. So simple and elegant. But obviously, the mathematical presentation is a little bit different, and from a sociocultural point of view, boy, there was a lot of argument about this in the early 1900s. So you've all kind of encountered this at various times in your education so far. I know you talked about it in high school biology. I know you talked about it in certainly in biodiversity and evolution, maybe even cells and molecules, that is Bio 1 or Bio 2 if you transferred here from another institution. And there's also a presentation in the laboratory manual, which is being used and gives the whole idea is that in order to figure out Hardy-Weinberg equilibrium and in order for it to maintain its truth, in quotes, there are a series of assumptions that have to be made. And I was very lucky when I was in France to meet and spend a little bit of time with Albert Jacquard, one of the first, actually, I shouldn't say that it was one of the first, but it was certainly one of the very important population genetics textbooks I read when I was in graduate school was a French translation of Albert Jacquard's textbook. Later on, when I was at Penn State, some of you may recall from my early introduction, I spent a mini sabbatical in France, and I was so lucky to be able to meet with Albert and have lunch with him before he passed. And one of the things that I so valued about Albert's view of population genetics, and specifically about his approach to Hardy-Weinberg equilibrium, was he looked at the assumptions in a slightly different way than gets presented to you in introductory biology courses. So I want to present it to you as if we were in a graduate program with a deeper understanding, rather, of definitions and certainly the mathematical background behind the ideas. So generally, populations, and in lab, when we talk about breeding fruit flies, when we talk about breeding yeast populations or mold populations, we consider a population to be any group of interbreeding individuals. All right? Human populations, certainly interbreeding individuals. But when we think about it from a genetic point of view, populations are actually very, very complex. First of all, only certain members of the population are able to reproduce. Okay? Obviously, certainly if we think in terms of animal populations, reproduction cannot occur until the individuals achieve sexual maturity. This is always true in any life cycle of any eukaryotic organism, okay? So we have to wait for sexual maturity to occur. Some percentage of the population is always not sexually mature. Further, some members of the population at a particular age has their ability to reproduce decline. So only certain members are actually able to reproduce effectively. Second of all populations have immigrants and emigrants, meaning that people are coming in and outside of the population. So they're bringing alleles into the population. They're also having alleles exit the population due to immigration. Again, really important, and those individuals still contribute genes to subsequent generations, certainly contribute to differences in gene frequencies in subsequent generations. Finally, unequal birth and death rates always cause the population size to vary. So when we think about a population, we have to think about defining our population in very, very strict terms. So what I'd like to do is reorganize the basic assumptions of Hardy-Weinberg, all right? And the first thing we have to do is define what we mean by a population. So a population is a group of individuals that belong to the same generation whose members only breed with one another, okay? There's no cross-generation breeding. Individuals cannot leave the population, nor can anybody come in. I always have this image in my head of a population isolated in time and space. They're in a little bubble, okay? Only individuals, and that bubble basically gets individuals removed from it periodically so that when the new generation comes along, the older members are gone. So we really have this situation where we've got no migration, individuals from different generations can't interbreed, and they're in this little bubble, or, if you'd rather think about it this way, in little fruit fly vials. So our fruit fly vials are basically designed to imitate Hardy-Weinberg equilibrium requirements of a population, because that makes it very, very easy to track inheritance, because everything is, or as much as possible, controlled. Once we define our population, then we can talk about alleles. And so what do we say about the alleles themselves? We say mutation does not occur. There is no mutation. All alleles stay the same generation to generation to generation. Again, it's a simplifying assumption. We also say there is no selection. Now, this is one that confuses, I think, beginning genetics students, is because they have a tendency to think of selection occurring at the level of the diploid organism. And to some degree, that's true. But that selection is dependent upon their genic structure. That is, it depends on the contribution of individual alleles to their fitness value. So when we think about selection, it really has to do to the value of a particular allele to the fitness of an individual. And fitness, remember, is in terms of not that individual surviving, but in terms of Darwinian fitness. That is the ability to reproduce, or reproductive fitness. Individuals can produce different numbers of offsprings just because they may be a little closer to the feeder, they may get more food, they may get into competition, which draws them away from reproducing, or they have different reproductive potential because of different alleles within their genome. So we have to say now there is no difference in reproductive potential associated with having different alleles. So the number of offspring any individual produces is completely independent of allelic structure or genotype at any locus. So this is really an important element. No selection. Finally, we can talk about genotypic structure. And genotypic structure, again, a little bit difficult to think about from an elementary or beginning genetics point of view. Genotypic structure is all about gametes. In other words, we've been talking about populations, we've been talking about alleles, and now we need to talk about genotypes. And how do we form genotypes from alleles? Alleles, of course, are considered in a haploid structure. Haploid to diploid genotypes. Remember, genotypes are diploid, the combination of two particles of inheritance. What we're really talking about, how do gametes combine? How do these alleles combine to form genotypes? And ultimately, what we talk about gametes, we're talking we need to figure out how the gametes meet. What are the different ways that gametes can meet? And ultimately, we have two assumptions within the idea of genotypic structure. And the first is that all mating must be genetically independent of one another. That is, alleles do not influence mate choice. This is called non-assortative mating. Okay? But even if there's non-assortative mating, if you have too small a sample size, there might be a change in gene frequency if you have small populations. And this is a little bit of a misnomer, because the real key is if you have a small population size, you're getting a small number of gametes produced. So what we have to assume is that the population is so large, it allows for infinite gamete production and therefore an infinite combination of gametes. So the population itself does not have to be infinite. This is a mistake in a lot of elementary or beginning biology textbooks. But the population has to be large enough that it allows for infinite number of gametic combinations. It's a really easy summary way and shorthand way of saying infinite population size. Obviously, infinite population size has infinite gametic production. So it does fulfill it, but mathematically it's important to recognize that we're really talking about gametes and not the population itself. So a little bit different. If you go back through these slides or through the video, you can kind of see how the organizational structure is a little bit more logical. And if you take advanced genetics courses, some of you maybe going to grad school and taking advanced genetic courses, and it'll be really important to understand this organizational scheme to do better in a population genetics course or an advanced bioinformatics course. Really important at this point in time. So it's nice to have an opportunity to talk about the complexity. Here's just a couple of very simple examples. You remember our M and N blood types. The M and N blood groups are controlled by two codominant alleles. Slightly different nomenclature we used in the past, LM and LN. In this case, it's just listed as AM and AN. And we have a situation here, again, by Race and Sanger in the 60s. This is a classic example. We see we've got 363 M individuals, 634 MN individuals, and 282 N individuals. There are a total of 1279 individuals, and of course, the relative frequency of those individuals within the population. And we can calculate this very quickly. We can look at it and say so the allelic or genic frequency of P, or the LM, allele, is 363. Why? Because there are 363 individuals. Half of the 634 MN individuals will have half of the alleles are M, and we're dividing by a total of 1279. It should be pretty obvious to some of you following along. There are other ways to do this. You could do it by saying, well, these are diploid individuals, so we have to do 3 times 363, right? And that'll give us the total number of LM alleles in the homozygous CAP-M individuals. Then we're going to add 634, right? Because that's the total number of LM individuals in the 634 individuals. we're going to take this entire quantity and divide not by 1279, but by 2 times 1279, because that is the total number of alleles in the population. Now, mathematically, these two equations are equivalent. We can then use the same math, figure out how many Q or Ln alleles there are in the population, use the same math, and we see 53 and 47. And then, of course, I'm going to challenge you to calculate the genotypic frequencies. We know, of course, that what you're going to do is plug our Q and P values into the Hardy-Weinberg equilibrium, P squared plus 2PQ plus Q squared. And of course, very simple to do, just a little bit of practice. Go ahead and pause and take a moment to do the calculations yourself and find out if the calculations match the actual frequencies of the individuals within the population. Once you've done that, one of the things reading Jacquard's book back in the 80s did for me was actually finding the contribution of an Italian population geneticist, this is Di Finetti, at creating Di Finetti's parabola. Something in the back of your mind, integrating your old algebra and trig classes from high school. I know it was a really long time ago, but if you think back, any time you have an equation with a variable squared in it, if you were to plot the equation, what will that graph look like? And of course, most of you know by this time, even if you're just kind of reading along, anything with a squared variable when plotted turns into a parabola. And so Di Finetti did a phenomenal job in recognizing this and was the first one to plot a parabola. And you can kind of see here, he talks about on the x-axis, we've got the p-values going from 0 to 1 in a left to right direction. Q goes from 1 to 0, right, because they are inversely related if you only have two alleles at a particular locus. And he is able to plot the parabola for the homozygous small a, homozygous cap A, and the heterozygote cap A small a. And you can kind of look at the parabola just like this. I love this because it is a lovely graphical proof of Hardy-Weinberg equilibrium. There are multiple ways to achieve a mathematical proof of Hardy-Weinberg equilibrium to show that P plus Q equals 1, and then, of course, the frequency of homozygotes and heterozygotes attain the values we can calculate. And the proofs are very elegant. You can use Hardy's original proof. You can actually do a fairly simple proof using matrix algebra, although you have to know matrix algebra, which some of you may. Most of you probably have not taken matrix algebra. Or you can use the graphical proof, and that's one of the reasons that I really like this. And you can really see this spot right here. If we take a look, if we were to draw a line, and I am so bad at drawing a line. All right, that's better. That's better. That's better. That's better. Okay, this is why you need computers to draw a line. Oh, forget it. I was going to try to erase my... Anyway, pretend this is a straight line. And if you look at where the frequencies of the two homozygotes intersect, where do they intersect? 0.25. And what does that tell us about the frequency of the heterozygote? 0.5, and it shows us very nicely 1 to 2 to 1 ratio. Okay, 1 to 2 to 1. Lovely. And you can also see how you can calculate or at least get the ratios from the graphs by looking at any particular combination. Just a lovely graphical proof. I'd be remiss if I didn't show you Jacquard's take on the parabola. And basically what Jacquard did was he put a simple XY type graph into a triangular graph pattern and really added a few definitions here, and you can look at the definitions on the lower left hand of the slide, and that is we've got D, which is P squared or the double dominant, H, which is are the hybrid individuals, 2PQ, and R being the homozygous recessive individuals, Q squared. You can also do the same thing by looking at this individual parabola and by dropping lines that are perpendicular to each of the three axes, you can actually calculate the individual genic frequencies for each of the different alleles. So very, very clever, but I think it's also very complicated. So it's not really, I like to show it because it's from the textbook, but it's a little bit more complicated. I think if we were to take a peek at D. Finetti's parabola, we would all agree it is it is a much more elegant presentation. So everything we have implies diploidy, right? We said diploid organisms. And that means that the traits we were looking at were autosomal traits. There's no variation in frequencies between P and Q for males versus females, right? Because all males and females in eukaryotes all have the same autosomal complement. Okay, so for example, using not the best of examples, if we are looking at tongue rolling and if it's got a frequency of 0.8 in males, it's also 0.8 in females. But what if the condition was X-linked? If we think about hemophilia as an X-linked recessive condition, our possible male genotypes could be a non-hemophiliac male or a hemophiliac male. Now notice we do have to remember to keep our X chromosome and our Y chromosome indicating these are males. If we take a peek at this, remember that when we present genetic information for a sex-linked gene, we're really indicating two different genetic conditions. The first and foremost, of course, being the sex of the individual, XY equals male in the mammalian species, and certainly then looking at the individual allele for the gene we're talking about, in this case CAPH or normal blood clotting, and small h for hemophilia. So if we're thinking about Hardy-Weinberg, there is no P squared, there is no Q squared, because there are no diploid individuals for the X chromosome. They only have one. So male genotypes are just simply P and Q. There is no P squared. Females, on the other hand, because they are diploid or homogametic, they are homogametic. They have two of the same sex chromosomes. Males, of course, are heterogametic. males are heterogametic. Females, of course, homogametic. Heterogametic, they have two different sex chromosomes. All right? So females can fit all three Hardy-Weinberg genotypic frequencies, p squared, 2pq, q squared. Pretty simple. So if we know that the frequency of hemophilia in women, is 16%, calculate all the other percentages. Well, if we know this is 16%, I'm going to let you go ahead and pause here, calculate the other percentages, which means that you're going to need to calculate what? You're going to need to know P and Q in males, and of course in females, you're going to need to know P squared plus 2PQ, sorry about that, plus Q squared, and calculate all those percentages. So take a moment, think about it, and calculate them. Hopefully you all had a chance to do it. What we're going to look at is basically 16% is Q squared, right? It is the homozygous recessive individuals, which means if 16% is Q squared, the square root of 0.16 is 0.4, which means we know that Q equals 0.4 and P therefore must be 0.6. Why? Because P plus Q equals 1. So 1 minus 0.4 is 0.6. So we know then, if we want to look at females, we know that 2PQ, 2 times 0.6 times 0.4, is 48%. These are the heterozygotes. P squared individuals, 36%. Okay? Really simple. Just got to take your time and work your way through the math. Of course, what about the guys? Well, as we noted a couple of slides ago, the afflicted individuals, or hemophiliac men, are the same as Q, which is 40%, and non-hemophiliacs are the same as P, or 60%. So, pretty straightforward calculation, just a slightly more difficult example. This all gets to fit in when we look at pedigree analysis, and certainly when we think about genetic counseling for individuals with sex-linked phenotypes. You can see how this would really complicate your view of conditional probabilities. One of the fun things to look at, and I do apologize for this graph a little bit. This is an old Microsoft Excel graph. I think I did it on an old original Mac. I have to redo this. And basically, if we start out looking at the P and Q frequencies, if we cross two individuals, one having an individual of 100% and the other having 0%. So we're basically creating a situation where there are hybrids. Because males are heterogametic, you can see that there is a skewing of the average, which we would normally expect to see 0.5 in a hybrid, right? 1 to 2 to 1. Half of the individuals are hybrid. We see the skewing towards what? 2/3. Why? Because there are 2 X's in males and only 1 in females, so we see a skewing of the average to 2/3. So just a cute little detail about how crossing can be affected by sex-linked genes. So pretty simple. X-linked recessive. The value for Q is the same as the frequency in males. In X-linked dominant conditions, the value for P is the same as the frequency. A final example, These data from the scarlet tiger moth, work done in the early 70s by great population geneticist D.B. Ford. And the null hypothesis is that the population is in Hardy-Weinberg equilibrium. The alternative, of course, is that the population is not in Hardy-Weinberg equilibrium. So how do we define this? And here are the data that Ford calculated. We've got white spotted individuals, the heterozygotes, and little to no spotting 5. You can turn around and calculate the allele frequencies. You can check my math if you like, and you can see that Q equals 0.046. What then would be the Hardy-Weinberg equilibrium? You go ahead and calculate those. How? P squared, 2pq, q squared, and you can double check the percentages with the numbers above. The fun thing is, of course, how do we know? In other words, how do we know? How do we define whether or not our null hypothesis is correct? And of course, the answer is our chi-square analysis. So how do we do our chi-square analysis? We look at the values for each of these individuals. So these are our observed minus expected. We create a chi-square table. We have a chi-square value of 0.83. There is only 1 degree of freedom because there are P plus Q, right? So there's only 2 possible alleles. And if we look at what our significant chi-square analysis would, or rather our chi-square statistic is 3.84, and of course 0.83 is not even close to 0.83. So we do not reject the hypothesis that the population is in Hardy-Weinberg equilibrium. So I wanted to just show you, it is not sufficient just to look at the values for P squared, P and Q, and arbitrarily say, well, they kind of match the data. Okay? Could we look at our observed ratios from our Punnett squares and say, well, they kind of match? And the answer is no, of course not. You have to do a Punnett square. Punnett square calculations are right here, and we're able to put them together and specifically show that we do not reject the hypothesis, meaning that we accept these individuals are in Hardy-Weinberg equilibrium. So a pretty simple view of how we go ahead and look at Punnett squares. Should be very easy for you to apply some of these ideas to the lab portion if your instructors are using the population genetic worksheets, and if not, this gives you enough experience. I also want you to start thinking about how we organize our definitions, because mathematically it's very important to keep our definitions both genetically and mathematically consistent. And I think that's the most important part of the early lecture. All right? Hope you guys are staying safe. Hope you all are well. And we'll see you next time.